Alright guys, today we're going to talk about mitosis. So with mitosis, please make sure that you write down any clarifying questions that you have. I do expect you to come with a clarifying question, and I also expect you to bring one interesting fact from mitosis, and please write those down because I'm going to collect those. Okay, so I want to review really quickly. Within the cell cycle, we've reviewed in class that we have two parts basically to the cell cycle. We have interphase, in which you've studied interphase. It's really the longest part of cell cycle, and it is all about cell growth. So we're looking at spending lots of time to get our cell prepared for mitosis, and mitosis is where we're going to see cell division. And so only in this small window will cells begin to divide. At other times they're kind of shut off from the idea of cell division, and they're preparing for that cell division. So mitosis is simply the division of the cell nucleus. And then we're going to see toward the end of the cell cycle cytokinesis, and cytokinesis is kind of a separate stage of cell division, and that's where we actually have the division of the cytoplasm, where we can actually get two separate cells that are not connected anymore. So a few important vocab terms that will help you understand mitosis. We have chromatids, and chromatids, remember, are taking a look at the chromosomes. So if I have, this is kind of how I typically draw my chromosomes. And so if I have, they're paired together. So here's a chromosome. Here's a chromosome. We call them, when they're connected like this, we call them sister chromatids. And then we have centromeres. You're going to see centromeres in the areas where each pair of these chromatids is joined. So I shouldn't have deleted that picture, but right in the middle, that area, that region is called the centromere. And then we have centrioles, and centrioles you're going to see within the cell during mitosis, and it's these structures at different poles of the cell that are within the cytoplasm, and they help to organize the spindles that help pull the chromosomes apart. And then the spindle, like I was saying, they're these microtubule structures. They will look like pieces of string in diagrams that basically help to separate the chromatids. So let's start with the first part of mitosis, which is prophase. Prophase is when we begin to see the chromosomes condense and become visible. So if you're looking at slides under the microscope, and you see the darkened region within a nucleus, and you can kind of see, that structure right here. That's what we're looking at. We're actually able to see the chromosomes because they have condensed and they have become visible. During this time, we're going to see those centrioles, those structures that are going to help us in the division. They're going to start to move to opposite sides of the nucleus. And then they help us to form that spindle. And so those spindles begin to form, and they're going to help us in the separation of the sister chromatids. And lastly, we're going to see the nucleolus disappear and break down, and we need this to occur, right? You see how it's like this dashed line kind of going around. Well, if the nucleolus does not disappear and doesn't break down, then the chromosomes, the DNA, cannot, be split up. It can't be broken down. So that has to occur. Okay, now we have the second phase, which is called metaphase. And when I hear metaphase, I think middle. So the M's kind of match up. So metaphase is middle. And that's where we're going to see our chromosomes line up along the center of the cell, and specifically the centromeres, which are those regions that are connecting the sister chromatids together. They actually line up right along the middle. Okay, and then during this time we see the spindle fibers, which are now in green. They begin to reach out and go and connect to those centromeres of each chromosome. And then we have anaphase. Anaphase, we begin to see the separation, the pulling apart of chromosomes. So these spindles have actually attached to, the centromeres, and that's where they're pulling those apart. And so now we are beginning to divide those chromosomes, that genetic information. And we see the chromosomes that will separate and basically get pulled toward opposite poles of that cell. And then the last phase of mitosis is called telophase. Telophase is when we begin to see the chromosomes get tangled, and as chromatin, they begin to not be condensed anymore. The nuclear envelope will have to reform because we're now preparing for cytokinesis, which is where we are getting the cytoplasm to basically split. And so we see the spindles break apart. The nucleolus then becomes visible again, and we begin to see two daughter cells that are pretty much the same. They may have a little bit more genetic variation, which we'll talk about later, but we begin to see two cells form. And so lastly, the other part of cell division is cytokinesis. That's where we're actually splitting the entire cells apart. And so the cell membrane will actually draw in until that cytoplasm pinches off into two equal parts. And so you could see here kind of this little bump right here, this splitting. If it's an animal cell, we call it a cleavage furrow. Okay, it's just a fancy name for animal cells. And with plants, their membrane is not flexible enough because plant cells are pretty rigid because of that cell wall. And so what you do see is the development of what is a cell wall. So this line right down the middle, I'm going to put some green right on it. That's going to be the cell wall. So you'll eventually see it totally separate like that. Okay? And we call this a cell plate instead. So you have cleavage furrow for the animals, and then plants have a cell plate. And there's a couple of links. I'm actually going to put these up for you guys, like through the pre-AP announcements or something. But if you want to look at these and kind of refresh yourself on mitosis, you can. All right, that's it.